Okay, folks, so this is video 2. In the previous video, we covered atoms, subatomic particles, different types of bonds, polar versus nonpolar molecules, hydrophilic versus hydrophobic molecules. Now we are on to pH. So we're going to talk about pH using this one single slide here. And then we're going to get into the different types of organic molecules. All right, so pH, where do we begin? Number 1, pH is basically a measure of how much hydrogen ions are in the solution. All right, now remember what hydrogen is. If you look back, we mentioned early on that hydrogen is an electron and a proton. That's it. It has no neutrons. So it's one hydrogen, one proton. But that's if hydrogen is neutral, because they balance each other out. If you are an H plus, a hydrogen ion, or more specifically a hydrogen cation, you must have lost that electron, right? There's no other way to become H plus. If you're a proton and an electron, you must have lost the electron. So the other name for H plus is a proton. The reason why I make sure I'm clear with this is if let's say you're in nursing school and they're talking about pH, and that nursing instructor keeps referring to protons, 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 but you only learned H plus, hydrogen ions, all right? You might not get it because you don't realize they're the exact same things, all right? So if you see proton, it's the same as a hydrogen ion. Okay? And the more of these you have in a solution, the more acidic the solution is. But here's the trick. The more acidic the solution, the lower the number. All right? That just sometimes gets students. All right? So I'll say it again. The more hydrogens you have, okay, the higher the concentration of these, the more acidic you are, but the lower the number on the pH scale. If you have equal amounts of hydrogen ions and hydroxide ions, which are OH negative, you are completely neutral. And neutral on the pH scale is 7. All right? So here's the scale. The pH scale goes from 0 all the way up to 7 and then up to 14. 7 is neutral. All right, so pure water is neutral because you're going to have the same amount of these as you do these. All right, now you're going to have a lot of H2O, right? But sometimes that molecule breaks into hydrogen ions and hydroxide ions. But there's going to always be an equal amount of those. All right, so then it's kind of, if you're looking at other solutions, it's basically what's the ratio of these to these. If you got a lot of these hydrogen ions, you're going to be acidic. So you go down. The more of those you have, the lower the number. All right, so down here is highly acidic. Stomach acid is very acidic. Battery acid is super acidic, but it has a very low number on the pH scale. So more acidic, lower the pH. If you go above neutral, you got less of these, and you might have more of these hydroxide ions. But the actual definition of pH has to do with these hydrogen ions. So if you have very, very low numbers of these, you're more basic. All right, and more basic means the number is higher. Okay, so that's the pH scale. All right. Now, what makes you an acid then? If you're an acid, you're a molecule that donates a proton or hydrogen ion into the solution. All right, so notice HCl, that's hydrochloric acid. That's basically the acid in your stomach. When it's in solution, notice what it does. It gives off or donates or dissociates into this hydrogen ion, aka proton, and this chloride ion. So that's the definition of an acid. It gives off these hydrogens because now you're increasing the number of these hydrogens, and therefore your pH is lower and you're acidic. Now, a base will remove protons from the solution. So a base is often called a proton acceptor. So like potassium hydroxide, when you put that in solution, it forms this hydroxide ion. Well, what does that do? Well, that will pull out hydrogen ions. So if there's hydrogen ions in here, it'll bond to that hydroxide ion. So it basically removes that hydrogen ion from the solution. All right, and that's what a base is. Salts are just two ions, bonded together that dissociate when they get into water. So like potassium chloride is considered a salt. Sodium chloride is a salt. All right, so that's kind of another type of inorganic molecule. These are all inorganic molecules. All right, so we know what the pH scale means. We know what an acid is. We know what a base is. Buffers help to stabilize the pH. So like, for example, your blood has to have some buffers in it if we're going to keep it at, 7.35 to 7.45. So there's buffers in your blood. Notice on the scale, these are some example, solutions and what their pH is. Now, a lot of these aren't that important to us, except maybe for me, beer is very important. But other than that, what are some solutions in the human body you might need to know? Stomach acid is very low. So it's somewhere between pH 1 and 2. So stomach acid is very acidic. If we were to think about skin is about the same pH as beer. All right, somewhere between 4, 4.5, somewhere around there. Notice that urine is typically around 6. It's a range, so some people could have urine down to even 4 or maybe even up to 8, but normal urine is about pH 6. Milk is about 6.5. So people like to think milk is not acidic. It actually is, to a chemist. Blood. There's blood. Remember, blood was 7.35 to 7.45. So that's blood. Blood is slightly basic. Seawater, like the ocean, is alkaline. That's the problem with pollution, right? Pollution acidifies the ocean, which is no good. And then these things are all very basic up here. One other concept to make sure you understand pH thoroughly is each unit on the scale is 10 times. So the scale is what we call logarithmic. So for example, if you were to compare lemon juice and stomach acid, right? It looks like that's about 1 unit on the pH scale. But 1 unit is 10 times. So stomach acid is 10 times more acidic than lemon juice. All right, that's how the scale works. So minor changes in the number are actually very significant, right? So when we start looking at blood, right? If you think about blood, 7.35 to 7.45, that range, although it seems like it's very, very small range, you can fluctuate quite a bit in there. Because those units are, again, a single pH unit is tenfold or 10 times different. All right? So hopefully that clarifies pH, acids, bases, the pH scale. One other thing to remember about, the human body and pH, we're largely neutral-ish, right? Blood is fairly neutral. But we might use acidity to reduce bacterial growth. So again, the skin is acidic. It's like 4, 4.5. That reduces microbial growth on our skin. The vagina is acidic. That helps reduce, vaginal infections. Stomach acid kills bacteria in the foods we eat. So a lot of acidity will help reduce, microbial growth. Okay, so let's move on to organic molecules. Again, definition, carbons and hydrogens. So these are all kind of drawings of monosaccharides. So a lot of these organic molecules have building blocks, right? There's a single unit, and then you can put a bunch of them together. You can do that with carbohydrates. You can do that with proteins. You can do that with nucleic acids like DNA. So we want to know what the single units are called, all right, kind of the building blocks, and then what can you form from those building blocks. So the building blocks of a carbohydrate are called monosaccharides. All right, mono meaning one, saccharide means sugar. All right, so we also often call these monosaccharides as well as disaccharides. We'll call them sugars. All right, a lot of times they're going to end in OSE. So if you see a word that ends in OSE, 9 times out of 10 it's a sugar, carbohydrate. The most important one for us is right here, glucose, because that's what our blood sugar is. All right, so we can get energy pretty quickly from glucose. All right, and you'll notice if we were to count atoms, glucose has 6 carbons, it has 12 hydrogens, and 6 oxygens. That's kind of a single monosaccharide unit. Some other monosaccharides you might have heard of, fructose. It's found in a lot of fruit sugar, if you will. Galactose. Galactose is part of lactose. Notice lactose isn't on here. Notice sucrose is not on here, because those are not monosaccharides, they are disaccharides. So again, disaccharides are also, called sugars. A disaccharide has 2 monosaccharides fused together by a covalent bond. So you'll notice here we have glucose, looks just like the glucose from the previous slide. There we have fructose, all right, both monosaccharides. If those were to be glued together through a chemical reaction, you would form sucrose, all right? So sucrose is a disaccharide made up of 1 glucose and 1 fructose, all right? Lactose, we've probably heard of lactose, that's milk sugar. That has 1 glucose and 1 galactose bonded together. And then maltose is another very common disaccharide. It's basically just 2 glucose molecules. Now, if we were to consume these in our diet, all right, let's say you had some sugar, maybe you had some milk, our digestive system is going to break that bond, all right, and it's going to go the other way. Oops, previous. So we'll take sucrose, and we have an enzyme in our small intestine that does a hydrolysis reaction and breaks them into 2 again. And then we'll absorb these into our body and use them for energy. All right? So those are monosaccharides, disaccharides, and then we might have polysaccharides. Polysaccharides have a lot of monomers. All right? Usually glucose, but you'll have some other monomers in there. And the most important polysaccharide in the human body is called glycogen. All right? So glycogen is a bunch of glucose molecules all bonded together. And this is how we can store glucose. All right? So when you consume food, right, consume carbohydrates, those carbohydrates probably have polysaccharides in them, either from, bread or, plants. We're going to digest that down into glucose, absorb the glucose into our body, use the glucose for energy, or if we've got plenty of it, we're going to store that glucose as glycogen. That's done at the liver. That's one of the most important jobs of your liver, is to take glucose from your blood and convert it into glycogen and store it for later. Your muscles do it as well, but your muscles are going to eventually use that glucose for energy, whereas the liver is going to put it into your blood to make sure your blood sugar is high enough. All right. So if you remember when we discussed homeostasis, one of those things with homeostasis is blood sugar. So if our blood sugar is low, we have to do something to raise our blood sugar. Well, the liver takes the glycogen and breaks it down into glucose, and that goes into your blood. All right, so that's how we fix low blood sugar. All right, very important, glycogen. We're going to get to it a number of different times in AMP1 and AMP2, but it's a stored carbohydrate. Okay, this is just a kind of a table. You know, if you missed anything in the previous couple slides, notice this is table format. Monosaccharides, some examples. Disaccharides, some examples. Polysaccharides, some examples. Glycogen is often called animal starch because it's how animals store glucose. Plants have a slightly different form of a polysaccharide called starch. All right, so starch is very much like glycogen, but it's found in plants. Okay, next organic molecule are your lipids. And under the title of lipids, there's really three types we want to think about. We've got triglycerides, which we're going to kind of talk about on this slide. We've got phospholipids, and we have cholesterol. All right, those are all lipids. Rule number one, lipids are nonpolar. All right, so they don't like to be in water. They can't form polar bonds, or hydrogen bonds, I'm sorry, with water. So if you can't form hydrogen bonds with water, you are hydrophobic. All right, so let's talk about, most importantly, we want to know what a triglyceride is. So let's kind of look here first. All right, this is a triglyceride. How did they get that name? It's got 3 of something. Those are called fatty acids. So that's the tri part. And then it has what's known as glycerol. All right, this area of the molecule shaded in this kind of bluish color is called glycerol. So you got glycerol and 3 fatty acids, so it's called a triglyceride. All right. Now, what are these triglycerides for? Well, it kind of depends on the organism. We use them to store energy. So fat is mostly triglycerides. We use them, we can convert them into other things. We can convert them into other like hormone-like molecules. We can use them for a lot of different things. What makes triglycerides different from one another are the types of fatty acids. So just looking at this triglyceride, you'll notice that the 3 fatty acids are all a little different. This one is longer than this one. All right. This one has a big kink in it. All right. So these are 3 different fatty acids. So triglycerides, there's a lot of variation. All right. A triglyceride isn't exactly like all the other triglycerides. It all depends on what 3 fatty acids it has. Okay. Now, we've probably all heard of saturated and unsaturated fat. This is when we learn about that. All right. Now, you typically learn about that in nutrition. Like saturated fat tends to be bad for you, contributes to, heart disease and cardiovascular disease. Unsaturated fat is usually considered healthier for you. All right. But what at the chemical level is the difference? All right. And we're going to look at these two. All right. This one here, stearic acid, versus this one here, oleic acid. In fact, they're shown over here. Notice the difference. Jumps right out at you. This one here has a double covalent bond, which puts a kink in it. All right. So a double covalent bond means that these 2 carbons are actually sharing 2 pairs of electrons. All right. Now, let's think about first what saturated means. If you think about something that might be saturated, let's think of like a sponge. A sponge that is saturated is full. All right. It's full of water, let's say. So you can't get any more water into a sponge that is saturated. All right. Now, why did they call this a saturated fatty acid? It's full of hydrogens. So you can't attach any more hydrogens along this fatty acid molecule. All right, there's nowhere to put another hydrogen. So it is saturated. Here, because of the double bond, it's unsaturated. Notice you could potentially put a hydrogen here, and you could put a hydrogen there. So this fatty acid is unsaturated. Okay? So that's where they got the words saturated versus unsaturated. All right, based on hydrogen. Now, why is this one bad and this one healthier? Well, saturated fat tends to pack together really tightly, all right, and that can increase the risk of cardiovascular disease. Whereas if you have a lot of unsaturated, it doesn't really pack together. And in fact, you can tell if it's got a lot of unsaturated fats because it's going to maintain liquid at room temperature. If you've got a lot of saturated fat, it solidifies at room temperature. So like bacon grease is going to solidify because it's loaded with saturated fats. Olive oil is liquid at room temperature because olive oil has a lot of unsaturated fats. All right, so hopefully that helps you. This comes up in every biology class or chemistry class. So I try to take my time and help you understand where they came up with these words saturated and unsaturated. Okay, we've got phospholipids. So phospholipids, you want to remember a couple things. Number one, they're used to form membranes. Number two, they are both polar and nonpolar molecules. So what a phospholipid is it still has this glycerol. This is kind of where that glycerol is. It's got one fatty acid here. It's got another fatty acid here. It's missing the third fatty acid, so it can't be a triglyceride. But instead, it has what's called a phosphate group right here. So instead of a third fatty acid, it has a phosphate group. Now, the beauty of this molecule is the fatty acids are nonpolar. All right? There's no charges here. All right? No partial charges. The phosphate is polar. So now you have a molecule that has polar here and nonpolar here. If you got a whole bunch of these things, they form this membrane. Remember, if you're nonpolar, you kind of like to hang around other nonpolar molecules. So the fatty acids face each other. If you're polar, you're going to face the opposite way. All right? And that forms a phospholipid bilayer. All right? 2 layers of phospholipids that will then be very useful in forming the plasma membrane of cells. All right? So phospholipids form plasma membranes because they have polar phosphate heads and nonpolar fatty acid tails, we often call them. Okay? Now there is a word for a molecule that's polar and nonpolar. I don't have it on here. I should put it on here. It's called amphipathic. An amphipathic molecule is part polar and part nonpolar. All right. So phospholipids form phospholipid bilayers that are part of the plasma membrane of cells. All right. Next one are steroids. Steroids are lipids. Steroids are hormones. All right. So estrogen is a steroid, testosterone is a steroid, cortisol is a steroid. So these are all functioning as steroid hormones in the human body. They're all derivatives, meaning they're made from cholesterol. All right, cholesterol is also the same steroid structure. Notice this kind of A, B, C, D portion here, the 4 rings. Very similar in all of these molecules. That's what makes them considered to be a steroid. We'll get to all these things one day. I do want you to know that steroids are formed from cholesterol. All right, you have to have cholesterol to make steroids. And the good news is your liver makes cholesterol. You don't need to get cholesterol in your diet. You get enough from your liver. All right, so steroids are lipids. This is the summary slide. Again, fatty acids, triglycerides, steroids. Okay, so you can kind of look at this again. It kind of summarizes the last 3 slides. Next group are proteins. Proteins, couple things to remember here. The building blocks are called amino acids. So there's that word, or those words, amino acids. So amino acids are like monosaccharides are to carbohydrates. So to build a protein, you need amino acids. So that's first thing I want you to remember. Number 2, proteins have to fold. So they have these different structural levels of folding, if you will. So, this they call a primary sequence. That just means, okay, you got these amino acids. Then they're going to form either these kind of helical folds, or this is called a pleated sheet fold. So you eventually get this really nice folded up kind of string of amino acids. All right. So proteins must fold in order for them to function. All right, that's a very important phrase. Proteins fold in order to function. All right, it's all about their shape when they fold. If you do something to cause them to unfold or fold improperly, they're not going to work. All right. Some things that cause protein to unfold, heat will do it. So if you heat up like a steak, or you cook a steak, the proteins unfold. And then acid does it as well. So when protein gets into your stomach, it's going to unfold. And that makes it easier for us to digest. All right. What do proteins do? They do all kinds of things. They can be enzymes. They can be like hemoglobin, so they can transport, oxygen. Proteins do a lot of the work that's going on in your body. And that's what this kind of summarizes. So proteins can be structural, such as collagen. You've probably heard of collagen. A lot of collagen in your skin. Collagen is in tendons and ligaments. Keratin is in your skin and fingernails and hair. So those are all structural proteins. Regulatory proteins would be things like hormones and neurotransmitters. They can help contraction. So contractile proteins, when we get to muscle, actin and myosin proteins. They function in our immune system. So you might have heard of antibodies. Antibodies help us fight against foreign substances like viruses and bacteria. They can transport. Again, hemoglobin is a protein that transports oxygen and carbon dioxide in your blood. And then they can be what's called catalytic. That means they're enzymes. So they can do, maybe they're digesting your food. So digestive enzymes. They could be cellular enzymes, all kinds of things. So proteins do a lot for you. Okay, last category are the nucleic acids. They also have building blocks. The building blocks are known as nucleotides. All right, so there's that word, nucleotide. And there's 4 nucleotides in DNA. They're called cytosine, guanine, thymine, and adenine. All right, and those nucleotides form what's called pairs. So cytosine binds and links up with guanine. So when you're forming DNA has to be in what's known as a double-stranded helix. All right, and we've probably all seen a drawing of DNA at some point. This is that double-stranded helix. So notice the 2 strands are being held together by these nucleotide pairs. You're always going to have a cytosine attached to a guanine of the other strand. Or you're going to have a thymine attached to an adenine of the other strand. So these are what's often called complementary pairs. All right. You'll never have a thymine bonded to a guanine. Doesn't work. All right. So those are the 4 nucleotides that form DNA. That's the specific pairing in order to form a double-stranded helix. RNA, single-stranded, doesn't have this double-stranded. And instead of thymine, it has a different nucleotide called uracil, which I'm sure I mentioned that on a next slide. So you got DNA, you got RNA. You also have what's known as ATP, which if you've never heard of ATP, you've probably never taken a science class. ATP is our energy currency. So it's what we make from carbohydrates, what we make from, maybe we're getting energy from triglycerides. Anything that you consume in order to get energy from, that energy is in the form of ATP. All right, and that stands for adenosine triphosphate. So it's got this adenosine structure here, and then it's got 3 phosphates. And in those bonds between the phosphates is where you have a whole bunch of energy. And we'll talk about this at some point, but again, if you've never heard of ATP, you've probably never taken a science class. It's our energy currency. Okay, last couple things I want to talk about is transcription and translation. This has to do with basically how do we make our proteins? You know, how do we make collagen? How do we make hemoglobin? How do we make an antibody? You got to have DNA first, right? So DNA, right, is in the form of our chromosomes, right? DNA has to be converted into mRNA, all right? So basically, mRNA is a photocopy of our gene. All right? Our genes are in the form of DNA. We make a copy of it, all right? Now it's mRNA. It's a slightly different molecule. Now that process is called transcription. So you're basically transcribing the DNA. You're making a copy of it. All right, and that's shown here. There's the double-stranded helix. We're making a copy in the form of mRNA. mRNA, therefore, contains the same information of that gene had. But what we can do with the mRNA is we can move it out of the nucleus, and it goes to what's known as a ribosome. And the ribosome will read the information in that RNA. And that's known as translation. Get it? Translates the information in the mRNA in order to make protein. So the mRNA has the code, right? It tells the ribosome what amino acids we need to make that protein. All right? So again, if you were to take a biology class, you get into a lot more detail here. For now, you know, it's useful to understand how, you know, the human body, how cells make their proteins. Right? They got to have DNA. It needs to be transcribed into mRNA. And then the mRNA has to be translated in order to make the protein. Okay? All right. Here's the final summary slide, DNA versus RNA. You can kind of go through it on your own. Notice the big, a couple big differences is DNA has thymine, RNA doesn't. Instead, it has a different nucleotide called uracil. The other thing I want to mention, double-stranded helix versus single strand. So that's another major difference. Obviously, DNA contains our, heredity, our genetic information, if you will, our blueprint. And then RNA, we mentioned kind of what mRNA does, but there's a couple other forms, tRNA and rRNA. Those are also, again, used during the process of translation, which we're not going to get into details. All right, so that, I believe, is the end of this video. All right, this chapter I did a little bit longer length just because it's hard to find a good place to stop. So hopefully you made it this far, and hopefully some of this chemistry is coming back to you if you had it previously. If you've never really had chemistry, or it's been a long time, don't worry. There's not going to be it's not going to determine your grade in this class, but again, in order to understand some of these other things, it's useful to have some sort of foundation or background in some of this basic chemistry.